Have you ever wondered how scientists can actually read a DNA fingerprint? I mean, it all starts with a huge puzzle. You've got millions of molecules that you can't even see. So in this explainer, we're going to break down the really clever technique called gel electrophoresis that takes that invisible mess of DNA and turns it into a clear, readable pattern.
All right, so here's our roadmap. First, we're going to tackle the core problem, why we even need this. Then, we'll get into the electric racetrack they invented to solve it. After that, we'll assemble our gear, actually run the race, and finally, learn how to read that photo finish.
So let's start at the very beginning. Before we jump into the how, we really need to get a handle on the why. What was the big problem that scientists were trying to solve in the first place?
I want you to just picture a test tube. It's full of what looks like clear liquid. But inside that liquid are millions and millions of DNA fragments, all different sizes. You can't see them, you can't grab them. So how in the world do you sort them out? This exact puzzle is what sparked the invention of gel electrophoresis. So how did they solve this problem of sorting something you can't even see? Well, the solution is honestly kind of brilliant. They figured, hey, instead of trying to see the molecules to sort them, let's make them race. Okay, so the official name for this is gel electrophoresis. At its core, it's just a method that uses an electric current to push molecules through a gel. A good way to think about it is that the gel is like a microscopic obstacle course, and the electricity, well, that's the engine that's pushing everything forward. But hang on, why does electricity make DNA move at all? Well, it turns out DNA has this little secret weapon. It's got a negative electrical charge. This all comes from its chemical backbone, specifically these things called phosphate groups. And, just like with magnets, opposites attract. So when we flip on the power, that negatively charged DNA just automatically starts moving toward the positive end of our racetrack. Simple as that.
And here's where it gets really, really clever. The gel isn't just some open road. It's more like a thick, microscopic jungle gym. The smaller DNA fragments, they're like these little race cars. They can zip through the mesh pretty easily and travel really far. But the bigger, bulkier fragments, they're like these heavy-duty trucks. They get tangled up, they move way more slowly, and they don't get very far at all. And that simple difference in speed is what sorts the DNA perfectly by size.
Okay, so we've got the theory down. Now for the fun part. Let's assemble our toolkit. What kind of gear do we actually need to set up this molecular race? So every piece of this kit has a really important job. The agarose powder is what we use to actually make the gel. That's the racetrack itself. The buffer is basically a special salt water that lets electricity flow through the whole system. Our DNA samples are, of course, the runners. We mix them with a colored loading dye that does two things. One, it lets us see our otherwise invisible sample. And two, it makes the sample heavy so it sinks right into the starting blocks. And naturally, the power supply is our starting pistol.
Now, before we go any further, a quick but super important reminder. We're working in a lab here. Safety always comes first, so you gotta have the right gear, like gloves and safety goggles. No excuses.
Okay, the toolkit is assembled, safety gear is on, it's race time. Let's walk through it step by step. First up, we have to build the racetrack. We mix that agarose powder with buffer, heat it up till it's a clear liquid, and then pour it into a casting tray that has this special comb in it. As the gel cools down and solidifies, kind of like Jell-O, that comb creates these tiny little pockets, or wells. Those are going to be the starting gates for our DNA runners. All right, this next part, this is where you need a really steady hand. Loading the samples can be tricky. You've got to guide your pipette tip into these microscopic wells without poking a hole in the gel or spilling your sample everywhere. Trust me, it takes a little bit of practice to get right.
So, we take our solidified gel, place it in the tank, and cover the whole thing with buffer. Then very carefully, we use a micropipette to load our DNA samples into each one of those wells. Once all our runners are in their starting gates, we pop on the lid, connect the power, and bang! We fire the starting pistol. The race is on.
Now, listen up, because here's a pro tip that will save you a world of pain. You have to get the electricity flowing in the right direction. The golden rule in any lab is run to red. Remember, DNA is negative, so it's always going to run toward the positive pole, which is always color-coded red. If you get it backwards, your precious DNA sample will literally run the wrong way, right off the gel and into the buffer. Poof, gone forever.
This race isn't exactly a 100-meter dash. It's more of a middle-distance run. We usually apply somewhere between 80 and 120 volts and let it run for about 30 to 45 minutes. That seems to be the sweet spot to get a really nice, clean separation of all the DNA fragments.
Okay, the timer goes off, we cut the power, the race is over, but our results are still totally invisible. So now comes the most exciting part, revealing the photo finish. So the separation is done. The small fragments are down at one end, the large ones are at the other. But again, how do we actually see them?
Well, to make the invisible visible, we use a special fluorescent stain that grabs onto the DNA. So when we slide the gel under a UV light, that stain, and by extension the DNA, glows super brightly. What we see aren't single molecules, but these bright bands.
Each band is just a massive pileup of millions of DNA fragments that are all the exact same size.
Okay, so here's the crucial part, knowing how to read this thing. The starting wells are at the top, so the finish line is at the bottom. The bands that are closest to the bottom, those are your smallest, fastest fragments. The bands still hanging out near the top are the largest, slowest ones. And this pattern is everything. Think about a crime scene. You compare the band pattern from evidence to a suspect's pattern. If they're a perfect match, you've probably found your guy. That's the power of this technique.
Now, of course, in a perfect world, every gel looks amazing. But lab life isn't always perfect. If you don't see any bands at all, you might want to check your power supply or your DNA sample. If the bands look all smeary, you probably ran the gel for too long. And if they're crooked, you might have had some sneaky air bubbles in your gel. It's all part of the learning process.
And that's pretty much gel electrophoresis in a nutshell, a really simple but incredibly powerful technique that takes an invisible jumble of DNA and turns it into an organized, visible pattern. This opens the door to everything from forensics to diagnosing genetic diseases. So I guess the real question it leaves us with is this: now that we can sort and see our own genetic code, what mysteries are we going to solve next?